First of all, I would like to welcome you all, a very warm welcome actually, on behalf of the AT Society to this Inside AT Research webinar that we're having this evening. We've got a packed programme coming up over the next 90 minutes, which you can see on the screen at the moment. My name is Rob Deneen. I am a neuroradiologist, so that's a brain scan doctor based at the University of Nottingham and at the working with the team at the Paediatric AT clinic based in Nottingham. And I've been doing research into the brain changes in AT for probably about a decade now, and I've worked very closely with the AT Society. So it's a great pleasure for me to be invited to chair this webinar this evening. I should also mention that I'm currently the chair of the Scientific Advisory Board of the AT Society. And the Scientific Advisory Board is a group of researchers and scientists who consider how the AT Society should support research, particularly if there's any funding that we're offering for research. As the Scientific Advisory Board are vital in helping the AT Society to decide where they should invest that money. So that's my role within the AT Society. So the programme we've got for you this evening is quite a mixed programme. Some of it is an update on the really interesting research that was presented at the International AT Clinical Research Conference that was held in Loughborough in the early summer. And so we're going to have a recap of some of those presentations, but we've also got some speakers who are going to be presenting to you this evening work which wasn't presented in Loughborough, and it's a great pleasure to have Professor Shalom joining us from Israel, presenting work which he wanted to present in Loughborough, but unfortunately was unable to attend at the time. We've got about an hour and a half. The last 20 minutes is going to be a Q&A session. So if you have any questions at all, do please save them for the Q&A, or you can type them into the chat and we'll try and address some of those questions as we go along, or I can read them out at the end if that's how you'd like to do it. And we are going to try and keep to time. So I'm going to chivvy our speakers along if they go over time, because we really want to give you a chance at the end to ask any questions that you have to the panel. The final thing to mention before we get started is that we are going to be recording this session so that we can share it afterwards with people who can't make it this evening. So all of the talks will be recorded. The Q&A at the end, we are going to record it, but we won't share that because we realise some of you may want to ask questions, but we haven't got your permission to share the video. So what we'll do is we'll record it, but only for internal purposes, so that we can look at the questions and the answers that were given, and we can share those as text sometime in the future with people who can't make it tonight. But don't worry, you won't be featured on any video that's shared. It's just the speakers. So with that, I am going to hand over to our first speaker, Penny Jago, who I'm sure many of you may know. Penny is an emeritus professor of genome damage and stability at the University of Sussex, but she's also worked with the AT Society for many years. She's a trustee, and she's also previously been the Scientific Advisory Board chair for many years before handing over to me. So over to you, Penny.
Okay, so it's great delight to be here. And what I want to try and do tonight is tell you about the meeting, the research that we discussed at the meeting. It was a great meeting. There's lots of enthusiasm and I think lots of great things going on, but I don't have very long. So all I can do is try and go through some of the key points and try to tell you about some of the discussion that we had. And then if you've got any specific points, you can ask me questions. So first slide. So the first session was based on diagnostic challenges and especially newborn screening. So I'm going to start off with this session. I'm not going through in any detail because you've all been diagnosed, but there's points there. And if any of you have questions, then do ask me at the end. Next slide. So respiratory medicine was the next session, and that is, I think, is really gaining recognition as importance. Impaired lung function is a significant manifestation of AT, and around a third of AT individuals succumb to lung dysfunction. There was a lot of discussion about some of the causes, although I don't think we really know what that is yet. And there was a recognition that there needs to be constant monitoring of the lung function and very active management. And Yossi Shiloh later in this meeting is going to come to give us a specific talk, which he unfortunately couldn't give within this session. There were two talks on the use of ultrasound to assess swallowing and also for therapy. And swallowing kind of is related to the respiratory function. And I think there was general recognition that this is a tool and which can be really helpful in the future. Next slide. So cancer was a long session and a very important one. Around a third of classic AT patients develop lymphoid cancers before 18. And the older patients, especially AT variants, who tend to be the older patients, develop solid cancers. And the outcome has improved somewhat for lymphoid cancers, but really not very much for solid cancers. An issue is the sensitivity to chemotherapy, which traditionally has involved DNA damaging agents, and it's that AT individuals are particularly sensitive to. And 60% of treated patients suffer chemotherapy-associated toxicity. I think that's improving because we're now much more aware of this sensitivity to the chemotherapy. But the good news is that there are non-DNA damaging therapies emerging, and that was discussed, and there was real pressure put for that to be trialed for AT. We recognize the need for international collaboration and networking, and the AT Society to help that of setting up working groups to try and help with collaboration and networking. The aim, which is very much US driven, one aim is to have tumor groups to help provide advice and support to oncologists, but particularly those from countries where they don't see many AT patients with cancers, to try and aid treatment. And also that will enable more integration and understanding of what may be the better treatments. There was also discussion for improving diagnosis. MRI scanning, again, was recognized as a useful tool and discussion about that. And also the benefit of trying to identify a signature, which is some work being carried out by Ramsey Bowden. And that will all help probably with outcome because early diagnosis is good. Next slide. So we also had a session dedicated to talking about the mechanisms underlying progressive ataxia. That's a complicated area, and I'm not going into it in detail. But I want to tell you what I think was one of the highlights. We, for a long time, have recognized that, or have not recognized, but thought that the major lesion that is causing the progressive ataxia is a double-strand break, which we know that in your DNA, and that's the DNA molecule there with a double-strand break. And we know that ATM is involved in dealing with that lesion. But it's increasingly becoming recognized, and I think this was quite apparent at the meeting, that actually there are oxidative stress can result in your own cell's metabolism, can result in reactive oxygen species, which can damage proteins and DNA. And that can activate, is now known to activate ATM, and that may be the more relevant direction from which ATM is required in the Purkinje cells. But it was recognized that the progressive ataxia is likely multifactorial, and we discussed various mechanisms. Another area that was recognized to be really important was the mitochondrial dysfunction, and that mitochondrial dysfunction results in more ROS generation. So the DNA gets more damaged, ATM gets more activated, but of course we don't have ATM. And then there was also kind of separately from that mechanism was the role of ATM in energy generation and metabolism. And that's going to be covered in a separate session. So I'll come on to that elsewhere. Next slide. So we talked about various different avenues for treatment, and William Whitehouse is going to talk to you about the clinical trials that are ongoing. Here, I'm just going to highlight how it all fits together. Most of the clinical trials at the moment are trying to improve the clinical outcome when we don't have ATM present. And it's often ways of trying to target mechanisms. For example, there seems to be hyperactivation of an inflammatory response in AT, and in the neurons that can activate microglia, and dexamethasone is probably acting there. And then there's the mitochondrial function dysfunction I talked about, and various of the trials are trying to improve mitochondrial dysfunction. But there's another area where what we're trying to do is get ATM, a copy of the ATM gene into cells so as to allow ATM protein to be made. Antisense oligotherapy is one of the approaches, and that's going to be talked about by William. But what I'll tell you about here is how some approaches, which I think are exciting and are really progressing, how we're trying to deliver the ATM gene to cells. So this is gene therapy. And it's been challenging because ATM is a large gene, and the delivery, especially to the Purkinje cells, is very difficult. But there are three, I think, approaches that have really gone forward. One is something called split intine splicing. And basically, we just have two fragments of ATM in two independent vectors, put them into the cell, and the cell will splice them together and give you an ATM gene that can make the ATM protein. That's been successful in cells and in vivo in hematopoietic stem cells, and the challenge now is to get that to work in Purkinje cells in the brain. James Dixon talked about a nanoparticle delivery system, and that's working again well in cells and in the cerebellar region of mice. But what we now need to know is whether that's really affecting ATM function. And then Tagaki, a Japanese clinician, talked about something called the piggyback transposon system, which is a system that will allow the large ATM gene to be delivered. So I think these are all exciting approaches. They're not going to give a solution tomorrow, but if we can get them working, they really have a chance to be able to deliver ATM to patients and to cells. Next slide. There was then a session on how what do we need to do to try and get clinical trials optimized. And one of those areas within that session was how do we monitor progressive ataxia efficiently? And that's important because we have to know what the rate of progression is in untreated AT patients, so that if we have a clinical trial, we can see there's a benefit of treatment. One of the approaches discussed was the use of what's called wearable devices for monitoring AT. And these are mainly sort of watch type devices that allow continuous assessment of movements and falling and that sort of thing. And that's really looking quite good. And then another approach was the natural history study that was reported by Emily Petleaf, who's been working with William on that. Next slide. So finally, but by no means least, Anne and Jane Flint from the AT Society talked about the very important area of mental health and well-being. And they really stress that a close interplay between mental health and physical health is imperative to have to allow any treatments to become optimized. And I think this is an area where the AT Society are really pushing forwards and it's not been generally recognized as being so important, but I think it's really gained the importance of it is really gaining strength. We need to be able to integrate health and social care models. There was discussion about the type of stresses and problems that are felt by people in the area of AT, severe isolation as children and young teens, especially at a time when healthy peers are developing independence. And then the separate stresses for families, parents, siblings, and care workers. And all of those need to be kind of dealt with and supported in independent ways. Okay, so I'll finish there. Um, Hopefully, I'm not too much over or just about in time, and then answer any questions at the very end.
Wonderful. Thanks ever so much for that really nice summary, Penny. And I can tell you, according to my stopwatch, you're 3 minutes under time. So thank you ever
Oh,
so much for keeping to time. That's great. So yes, any questions for Penny, please do save them for the end. Okay, so our next speaker tonight is William Whitehouse. William has been many of you will know William in his role as the consultant urologist for many years at the paediatric AT clinic in Nottingham, and he's also an associate professor, clinical associate professor at the University of Nottingham. William, if I can hand over to you for your talk, please.
William, you're muted.
Classic. So can you do my next slide? Yeah, and the next one. So I was going to talk about this meeting that Penny's mentioned already. It was really exciting and inspiring meeting for the researchers. There are about 50 researchers from all over the world, from Japan, all over Europe, all over the UK, United States. So it was very exciting and inspiring for those guys, for all those guys, some more clinical and some more laboratory-based researchers. Next slide. And right at the beginning, we had an inspirational speech to get us focused from Alicia, who's a person with AT, which was very grounding actually and useful. And the clinical meeting finished and overlapped on the Friday with the AT family weekend. Next slide. So news about new treatments for AT. So for clinical trials, as Penny's mentioned already, we need to know the natural history. So we need to know what would happen without an intervention, and also to plot where and what age group to do what kind of intervention. So we need to know what things to measure, to look at, to see that what can improve with an intervention. And so that's the natural history. And Penny mentioned Emily, who's now in Birmingham, but she's working at the University of Nottingham on her PhD, looking at the natural history of all people who attended Papworth Clinic and also Nottingham Clinic over about 20-year period. So that's almost done now. She's locked the database, and it's just a matter of her completing that write-up. We also need to understand something about the biological mechanisms in AT. And again, Penny's alluded to that, and in particular to the double-stranded DNA breaks, which makes you susceptible to cancer and maybe to other problems like immunity. And she mentioned the reactive oxygen species, and she also mentioned the mitochondrial function. So those are all impacted further down a chain of causality, further down the chain really of the disease process from the mutation. So understanding biological mechanisms then gives rise to ideas about what might work for a treatment or hypotheses. And then there needs to be a small study to prove the concept that you got a new idea. Can it possibly work? Is it worth investing in a bigger trial or investing in experimenting on humans? And that's the kind of work that's been completed for some of these ideas, but not for everything. And then you need safety studies in humans, and then an efficacy study, so a bigger trial, what's called a phase 2 study. And if that's successful, then you need to go on to get a study that allows it to have the treatment to have marketing authorization or a license to advertise the drug for a particular indication. So that's a big phase 3 study. So they're all very expensive doing all those studies, and it takes many years. The cost effectiveness and affordability then in the UK is assessed by NICE, which is an organization some people will have heard of, which looks objectively at the cost effectiveness of a treatment and whether it's worth the NHS buying it and funding it. I think the problem for us in the UK is that the cost of some of the new medicines is very, very high because of the American market, and it's largely market forces that drive that cost. And then the cost is the same throughout the world, whatever the American market can take. And of course, the problem for us is that most countries can't afford those kind of costs. So in the UK, having an NHS means that we have bargaining power, and often these expensive drugs are costed following negotiations that are secret, and the NHS generally ends up paying us a reduced cost, but that it's secret, so we can't know what the actual cost is for some of these expensive new drugs. And then after the drug's available and being used, there's post-marketing surveillance, so that when patients are using it, they need to have review of what's happening, and that's when some rare side effects will be identified. Rare side effects might not show up in the clinical trials. So those are all the steps that new treatments have to go through. Next slide, please. And this is another cartoon of ATM sits in the middle of, well, not countless, but tens and tens, maybe 30 or 40 different reactions in the cell. That's key. And so there are lots of potential targets and lots of potential ways in which it can give rise to the problems that we see. And as we've already mentioned, the reactive oxygen species, that's too reactive in the cell. Energy loss, the mitochondria not producing enough energy in the cell, and the double-stranded breaks, the breaks in the DNA chain itself, leading to problems with replication, causing the cancers and some of the other symptoms. Next slide. So we had a session from world experts on these forms of treatment, to basically to either correct the gene problem or to try and bypass the gene mutation was the first thing that we looked at. And Penny's mentioned this antisense oligonucleotide treatment, or ASO treatment. So it's been pioneered in Boston with colleagues from Harvard University, Tim Yu and Claudio De Giustamo. And about 5 or 6 years ago, a first child aged 2 years, 10 months, was treated with a specific chemical that covers up the mutation that's in a splice site. So it's difficult to explain that all very briefly, but basically by kind of hiding the mutation from the apparatus in the cell so that the gene can then function more or less normally. And she's been having this treatment every 3 months, a lumbar puncture treatment of this ASO called antipexen. And so far, so good. You know, she's following a really benign course for someone who's aged 6 or 7 now. So that's very encouraging, and that's not been completely published yet. So that's really encouraging. The trouble with that is that her mutation is quite rare. So it's only a few people. I think it was only about 1% of the people in their pool that they looked for who had that particular mutation. Everybody got different mutation, and you would need a different ASO for a different mutation. The Cambridge group, that's Rita Horvath and Anke Hensig, and some of you will know, they're looking at working on a much more common mutation using the same technology, ASO technology. The mutation is relatively common in the UK, so it's sometimes called the English mutation. and it's one of the mutations that produces a milder variant of AT. And it comes in around almost 20% of UK adults with AT have that mutation. So they're pressing on with work setting up that study. I don't think they've started the treatment yet, but they've been doing a natural history for that. And then in Tübingen, in Germany, Matthias Sinofixik, he was very enthusiastic at the meeting and full of ideas. He wants to develop a platform so that he can get approval to do a whole host of different ASO trials all in one trial. Otherwise, if you cost several million US dollars to do one trial and there are loads of different ASOs needed, it's an intractable problem. So his idea is to try and do lots of different ASOs all in one trial with one set of approvals. So that would be great if he can pull that off. And then another way of assessing the gene would be to replace the gene, and Penny's alluded to this. And as she said, the Nottingham group, James Dixon's working on having a synthetic gene carrier to get a properly working ATM into cells, and that's worked in cell culture. And also he's got some data in mice, which is really encouraging. That's taken a few years to get that far from the initial idea, but it's all progressing well, and I think is a reliable avenue. Group in Naples are working on splitting the gene and getting it into the cell through two different vectors, and then the cell reunites it. So Penny already mentioned that Rossi Anguliana in Naples working on that, and the guy in Tokyo, Masatoshi Takagi, is working on a different system, different technology using this piggyback transposon helper system. So there's some encouraging data there. So all this getting this huge gene, 9 kilobases in, it looks like it may be on the cards in the near future. Next slide. Reducing the harmful consequences of the mutation. So I just wanted to mention the ERIDEX or EDSP trial that we've done in Nottingham and other sites throughout the world. So this trial was damaged to an extent by COVID, in that a lot of patients, particularly in India and poorer countries, couldn't get their treatment during COVID, and that kind of wrecked the trial from a technical point of view. Although the trial results were very convincing, if you just looked at the participants who actually got the treatment. So if you excluded those who missed several doses, it showed a clear benefit. The benefit wasn't so much a cure as just a slowing down of the decline of the AT in those young people, particularly in the youngest children, aged 6 to 10 years old. So it really seemed to, because of the technical aspects of the COVID and not everybody getting their treatment properly, the FDA, the American regulator, asked for another trial just to confirm that result. So that's fair enough. So that's called the NEAT trial. So that's been recruiting well throughout the world, including in Nottingham and London, and of course we don't have the results yet for that one, but that's work in progress. And the pictures show some of us working on the AT-NEST trial in Nottingham using the ODEX system. So blood's taken out of the child, loaded with dexamethasone, and then reinserted back into the child, retransfused back into the child every 3 or 4 weeks. And I was up there last week assessing some children who were in the NEAT study, and it's well tolerated with a very low dropout rate, even though it's quite an intrusive treatment. Next slide, please.
William,
Yeah.
can I just briefly interrupt and just say, would you be able to wrap up, because we're close to the end of the slot that we have? Thank
Will
you.
do. Sorry, I must have used those 3 minutes extra. So increasing the cell's energy, triheptanoin is a drug that's already used in other metabolic diseases. The Brisbane group discussed that, and their phase 3 trial has finished and has had successful results. So they'll be moving on to a more bigger clinical trial. Ataxia-Telangiectasia Intrabio is the company doing that study. That's a trial that's ongoing in several centres, including in Nottingham and Papworth in the UK. That's a much easier intervention. And nicotinamide riboside has been studied in Nijmegen and Oslo with some safety data, but not a full-blown randomized controlled trial yet. And then on the horizon, there's this drug almoxilolone, which has proved effective in Friedreich's ataxia and is an antioxidant, so that addresses the reactive oxygen species issue. Next slide. That's a publication that Penny and David Common did summarizing the meeting for those of you that want to read the more scientific, fuller details of it. Next slide. And thanks for giving me the opportunity to share a bit of what we learned at the meeting. It was very inspiring for researchers who need that kind of inspiration every now and then. Thank you so much.
Wonderful. Thanks ever so much, William. I know that clinical trials are something that always generates a lot of interest when we have these sessions, and so there may well be some more questions on this in the Q&A session at the end. Okay, so we're going to move on to our next speaker, and it's a great pleasure to be able to introduce Professor Yossi Shilo, who is Emeritus Professor at the University of Tel Aviv in the Department of Human Molecular Genetics and Biochemistry. And Professor Shilo has really been, I think, a leading figure in AT research for many years. In fact, I just quickly had a look on the big database of all the scientific research publications called PubMed. And if you put his name in and the words Ataxia-Telangiectasia, there are over 160 studies that he's contributed to. So, an amazing contribution to our understanding of the disease. So with that, I'm going to hand over to Yossi.
Thank you so much, Rob, and thanks for the kind introduction. It's a pleasure to be here tonight, and I'm still sorry I couldn't attend the meeting in the last meeting. But anyway, I was going to speak about several subjects, but one that it might particularly interest the parents of individuals with AT children and other people who carry AT mutation is the ongoing story about the health of AT carriers. As you know, the basic genetics is that carriers have one mutated copy of the ATM gene and the other one is normal. And when two carriers have children together, there's 25% chance in every pregnancy, independent of the other pregnancies. It's always this 25% chance of having a child who has two defective copies of the ATM gene, and these are the children with AT. And these days, prenatal diagnosis or pre-implantation genetic diagnosis are available for these families. So what about the carriers themselves? Historically, it's a long story, and we've been following the health history of a group of AT carriers in two Israeli communities for many years now. Because of the, other than the interest in AT itself, there's always been an interest in terms of public health in the carriers, who in some populations constitute about 1 to 2% of the general population. And it has been a very long study, and the good news is that we finally wrapped it up in a manuscript that was submitted and is under review. So hopefully in the not too distant future, all these data, which are big Excel sheets and long tables, will be out, but they are important. I'll try to summarize them as briefly as possible. I should mention that we started this study in You won't believe it, in 1998, 27 years ago. That's the nature of epidemiological follow-ups. And another thing which is kind of unique to AT in Israel in terms of the genetics, like I said, in individuals with AT, both ATM copies are mutated. And there are many, many, many mutations. It's always the same gene, ATM, but it can be mutated in many ways. It's a long gene, and it tends to be, as geneticists say, very polymorphic, meaning there are a lot of sequence alterations. Some of them are quite benign, but those who actually the geneticists call null alleles, because they totally inactivate or eliminate the ATM protein altogether, those are the mutations that lead to what we call classical AT, not the milder form. The problem in the proper way it should be always done was to identify carriers in specific populations and follow their health history over time. And the problem is that because there are so many mutations, there are over 800 AT mutations, not just ATM mutations, AT mutations, those null forms of the gene in the registry. Meaning that in many countries, individuals with AT indeed have two defective copies of the ATM gene, but the mutations on each copy are different. Geneticists call this situation compound heterozygotes. Okay, the end result is the same. There's no ATM or no active ATM, but the mutations on the two mutations on the two different copies of the ATM gene are different. The one from the mother and one from the father are different. And that's the situation in many countries. So because there are so many different mutations, and the way to identify carriers, those individuals who have one normal copy, healthy copy of the ATM gene, and the other one has the mutation, it's difficult to do it because you have to design a specific molecular test for every single mutation in order to do that. And that's usually impossible for the United States is a very good example. However, the situation in Israel is very different. It's different because AT was identified in very defined communities. Among the Jews, it's only Moroccan Jews, and among the Palestinian Arabs who live in Israel, who are citizens of the State of Israel, there are several communities, each with a single mutation that characterizes that community. Meaning the mutation occurred once in the history of this community and then continued to segregate in the community. And when two carriers of the same mutation had children together, that's actually what made us know that there is an AT mutation segregating in that community. So Moroccan Jews, all patients are what we call as geneticists homozygous, meaning it's the same mutation on both copies, and all the carriers in this community of 600,000 people, one out of every 100 individuals at random is a carrier of this specific mutation. So we could, for example, collect 1,000 random samples of individuals who are not associated with AT families at all and identify carriers in that group. Actually, we identified 10, which is exactly the proportion that we expected, 1 in 100, 1%. And the same is true for the Palestinian Arabs and for Druze and for Bedouins and so forth, which means that we can approach such community. In the case of the Palestinian Arabs, usually these are very well-defined communities, big kinships, usually all of them living in the same village or town, meaning they're all together, they all live in the same area, same standard of living, and we can identify carriers and non-carriers using a very simple test, which is what we did in 2 such communities, each community with its own mutation. By the way, one of them has the big, what we call deletion, part of the gene missing, which identified the ATM gene to us. It was that family. And I should acknowledge the enormous help that we got from these wonderful families. So anyway, we started that some 27 years ago because the literature was rich with suggestions that there might be something different with regard to the health of the carriers. And a lot of researchers were interested in that, but we were in, I think, a good position to do it in the right way. So we approached them, we genotyped them, meaning we determined who in that community is not a carrier and who is a carrier. And we interviewed them with the help of Palestinian Arab medical students who were the best, most appropriate people to approach these families, come into their homes and ask them all these questions, which are sometimes, very, very personal. And then we continued to follow them through the national registries of the cancer registry and the hospitalization registries. And in the beginning, which was 27 years ago, most of these people were quite young, but as they were advancing in age, we could start detecting some differences. I always say that the problem was that we were aging as well, the researchers. But anyway, the bottom line is that carriers are part of the general population with all the typical morbidities for various ages. But if you look at them as a group, you see in the more advanced ages, I would say as of the 6th decade of life, a little bit of excess of some chronic diseases like cardiovascular diseases and some cancers. But there's nothing that is outstanding compared to the general population. Statistically, when you do this kind of study, and we were following 105 carriers, and the controls, which were non-carriers, were some 800 from the same communities. Relatives, actually, all of them relatives, the same kinships. So all these details will be published soon, but there's one specific, very interesting and very important detail that we came across, which I always say when I discuss this is the first time we can tell the carrier something very, very definite, and it is simply refrain from smoking, because usually we sort of remove or neutralize the smoking effect. That's how epidemiological studies are done. But when we purposely look at the difference in terms of general health between carriers who smoke and non-carriers who smoke, what we found was not, as some people might think, excess of lung cancers, which is usually the biggest problem with smoking, not at all, but excess of COPD, those chronic lung diseases. I guess everybody has seen those heavy smokers who at some point start pulling with them oxygen cylinder. This kind of diseases. And this is actually reminiscent of the lung disease, the obstructive lung disease of individuals with AT, usually at more advanced age. So this symptom of the disease sort of exists in the carriers, but it is sort of masked. But if they provoke their lungs with smoking, then it comes up. It mirrors the obstructive lung disease of people affected with AT. So now we have a very, very definite and clear advice to the carriers: refrain from smoking. And other than that, just take good care of yourself like everybody else. So, and I'm really glad that we finally submitted it. You know, I've been discussing this study, partial details in meetings along the years. You know, we always told, we reported about the study in meetings, even when it was just starting and we didn't have very, very clear findings. So finally, it will hopefully be out soon, and I really hope that it will be a definitive paper about what's going on with the carriers. And I hope I didn't pass my time.
No, not at all. No, thank you ever so much for that talk. Yeah, so fascinating work, certainly unique, and as you say, I think it'll be a really significant contribution to our understanding of the health of carriers of the ATM mutation. No, thank you ever so much for coming and sharing that with us. Just before we move on to our next speaker, I just want to highlight that Susie has dropped into the chat a link to a video which summarizes some of the trials that William has already talked to us about. So do have a look at that link in your own time. The next speaker then, it's a great pleasure to introduce Dr. Anna Wilsden, who is a clinical geneticist working now in the paediatric AT clinic in Nottingham. So some of you may already have met her, or if you haven't and you do attend the clinic, you may well meet her on your next visit. So if I can hand over to you now, Anna.
Yeah, thank you. I'm just going to see if the slides will move because they don't appear to be. Oh, there we go. Okay, so I've got the joyous task of talking about the bits that nobody really wants to, which is cancer. And I know this is a difficult topic and not everyone wants to think about it, but I think it's actually really important. We all know there's an increased risk with AT, but what I'm going to talk about today is the carriers. So that's mum, dad, potentially siblings, the wider family, people who don't have AT but carry a single ATM gene change. So if you remember, it's changes in the ATM gene that cause AT. If you have two gene changes, one on each copy of your DNA, you develop AT. Having one gene change makes you a carrier, so you don't develop AT because you have that second working copy of the ATM gene change. And as I said, whilst it might be a difficult topic, I hope some of what I can say today might actually be reassuring for some of you and at least set things out so it's clear to you what screening might be recommended. So I'm going to start with breast screening. So we've known for quite a while that individuals with AT have a higher risk of breast cancer and they get additional breast screening from 25. But what about carriers? So back in the 1990s, there was some research coming out suggesting there was an increased risk of breast cancer for carriers, but some conflicting evidence wasn't quite clear. And then a bit more information came out and suggested that carriers had a 3 to 4 times the population risk of breast cancer. So we used to offer 18-monthly mammograms. And then a few years ago, there was a really big study that came out that looked at breast cancer risk from lots of different genes, not just ATM. But it confirmed that some ATM changes do increase the risk of breast cancer, but not all. And that's important to remember. It also showed that ATM is a moderate risk gene, so it increases your risk 2 to 3 times that of a person in the general population. It's not a really, really high risk gene like the BRCA gene that Angelina Jolie has, where people have surgery to remove the breast tissue. And many families with ATM gene changes will never have, the carriers will never have breast cancer. And you're still more likely to not get breast cancer than get it because of ATM. But because we know that there is this link with cancer for some ATM gene changes, we recommend that mums of all children with AT have moderate risk breast screening. So for women in the general population, your breast screening starts at 50 when you go into the National Breast Screening Program, but for carriers, so they were talking about the mums of children with ATM, with AT, you should start your screening at 40. Okay. But it's more complicated because we know not all ATM gene changes are the same, and it's only part of the puzzle. So what else can change your risk of breast cancer? And how does that change the screening that we offer you? And why, in some cases, do we offer to test other relatives to inform breast cancer screening and not other families? So there's 3 big things that can alter your risk of breast cancer and tell us whether you need more screening on top of that moderate risk screening starting at 40. So do you have a family history of breast cancer? What specific ATM gene change do you have? And what other non-genetic things influence our chance of developing breast cancer? So if we start off with family history, having an ATM gene change can increase your risk of breast cancer, but your family history often tells us just as much, sometimes even more than just knowing about the ATM gene change. So for example, if you have an ATM gene change, but no family history of breast cancer, that's really important. It probably means that some of your female relatives also carry the same ATM gene change, but they haven't developed breast cancer. So there might be other protective factors in the family or fewer additional risks in the family. And in this situation, where there's no family history of breast cancer, your risk is likely to be at the lower end of that increase due to ATM. And starting moderate risk screening from 40 is usually the right approach in this instance. But what about somebody who has a family history of breast cancer already? So I've highlighted 2 people in red here. If breast cancer runs in your family, especially if it's in close relatives like parents, siblings, aunts or grandparents, you might need extra screening. So more than just starting at 40. You have to remember though, cancer is really common, especially as we get older. So any family history we look at, there's usually a few cancers dotting around. And having a grandmother who had breast cancer at 85 usually doesn't change your risk much. But if you had 2 close relatives who had breast cancer in their 50s, maybe, that's more significant. And in that case, we might want to think about starting your breast screening earlier rather than waiting till you're 40. And in those instances, we'd recommend that you ask your local clinical genetics team or me when you see me in clinic for an assessment. So to summarize, if you've got no family history of breast cancer, you're probably fine to start your breast screening at 40. If you have got a family history of breast cancer, especially if it's in close relatives, we might want to think about starting that screening a bit earlier. Now, what about the ATM gene change itself? So your risk depends on the specific type of ATM gene change you have. Whatever type of gene change you've got, you still need to at least have that moderate risk screening from 40. But sometimes some people will need more screening, and the gene change can affect whether we recommend breast screening for your relatives or not. So there's quite a bit to cover here. And to explain why the type of change matters, I think it's helpful to go back to the basics and the science. So we know every gene, including ATM, makes a protein. Now, if I ask PowerPoint for a stock image of a protein, I don't get anything scientific or fancy, I get fish. But I thought, why not? We can use fish to explain this, so we'll give it a go. So think of the protein as a goldfish. When the ATM gene works normally, it produces a nice, healthy, fully functional goldfish. Different changes in the gene can affect how well that fish works, and that's why the exact change matters for your risk, because the research suggests that not all ATM changes increase your risk. So when there's a change in the gene, in the ATM gene, the DNA code is different. And in this case, the protein has stopped being made too early. So it's shorter than it should be. We've just got a fish head now, and it can't do its job properly. And this type of change is called a protein truncating variant. It's like chopping the fish a bit short. The fish can't swim properly because it's missing its fins and its tail. And in the same way, the protein, the ATM protein in the body doesn't work quite as well. And we know that this type of ATM gene change is definitely linked to an increased chance of breast cancer, but the risk is still just moderate increase. But that's why we recommend the moderate risk screening that I've mentioned starting at 40. And in this case, we'd also offer the genetic test to your relatives so they can get extra screening if they need it, because we know the link with breast cancer is much more certain for this type of gene change, the protein truncating change. But what if the DNA change doesn't shorten the protein and instead just makes it a little bit different? So this type of change is called a missense variant. And these are not proven in the main to increase your chance of developing breast cancer. So when there's a missense change, instead of making a nice normal protein, the DNA change gives different instructions. The protein's there, but it just looks different. It's a bit like here where we've got a fish which we wanted to have fins but has ended up with wheels. It's unlikely to work very well underwater. But saying that, amphibious vehicles do exist. Might be no good for going down the motorway to work or doing the weekly shop, but maybe it could do a little bit of its job. Maybe it could ferry some tourists across the river. So the effect in the protein can be quite difficult to predict in this instance. It might mean it doesn't work at all, as we see in many cases of AT. And so sometimes this missense variant does have the same effect as that shortening variant, the truncating variant. It just doesn't work. So you think, well, surely this should have the same cancer risk as a truncating variant. Both types cause AT in the child, so shouldn't we treat them the same way? That's not what the research is telling us. And actually, when they went back to look at a bigger cohort to say, to see are missense variants like this associated with an increased breast cancer risk, it still didn't show a clear link. So do women with missense changes need extra breast screening? Well, that's something we still don't have a definite answer for. And this is why it gets so complicated discussing breast screening. For now, mums of children with AT who have missense variants and protein truncating variants, we would still say you can have moderate risk breast screening from 40. And for the missense variants, that's probably us being a bit overcautious. But for missense variants, we wouldn't offer to test the wider family to inform their breast screening. And this can feel quite uncomfortable when we're saying different things to different people in the family, where the same ATM gene change is being passed on. So how do you, as a mum of a child with AT who's been offered extra breast screening, how do you explain to your sister that actually we're not going to offer her a genetic test and she wouldn't be offered additional breast screening based on whether she's got the ATM gene change or not? This is something that's been the subject of a lot of debate, and there are a few reasons behind this decision. So partly because the evidence for missense variants causing breast cancer isn't quite clear. We don't want to have lots of women having unnecessary anxiety and potentially unnecessary mammograms when we haven't shown that there's a really clear risk for some of these changes. So we restrict it to a smaller group of people, just the mums. We know that having an ATM gene change does not increase your breast cancer risk by a huge amount like some of the other genes, like the BRCA genes, where the risk is much, much higher. And the other thing we don't really know at this stage is whether breast screening for women with an ATM gene change carriers actually improves things in the long run. We don't know that finding a breast cancer earlier with screening saves more lives than if we just waited and the woman found a lump and was found to have breast cancer that way. So there's lots of work ongoing at the moment to clarify the situation. And this is something that's going to be under continual review, and I'll review it every time I see you in clinic. It also doesn't mean that relatives can't have screening. If there is a strong family history of breast cancer, they might still be offered breast screening based on that alone. So I've talked about how helpful the family history is to work out someone's risk, and higher risk women will hopefully be picked up this way. So to summarize that very confusing, slide's not moving. There we go. very confusing situation. If you've got a truncating variant where only the fish head is made, you get moderate risk screening at least. And your relatives will definitely get screening if they have the gene change. For missense variants where you've got the fish bus combination, the risks aren't quite clear. We would offer the mums moderate risk screening from 40 at least, but we don't offer to test and screen relatives. To make it even more
Anna,
complicated,
sorry, can I just ask you to move towards the end?
speed up, yeah. So to make it more complicated, there is one more additional gene change that's a missense change that we know is associated with a higher risk, and we would look at that with you and advise earlier screening. So what else increases your risk? Well, lifestyle is actually really, really important. So I've shown here we've got DNA for the ATM gene change and fast food for bad lifestyle. So someone in the general population has a 10% risk of getting breast cancer. We haven't quite worked out the lifetime risk of breast cancer due to ATM, but it's probably somewhere between 17 to 30%. Now, if you are overweight, that actually increases your risk of breast cancer by 30% and possibly more. Or having 2 to 3 alcoholic drinks a day can increase your risk of breast cancer by 20%. So you get the general idea that lifestyle is really important. So for women, the next steps are really to either discuss with us in clinic or ask your clinical genetics team. I'm just going to briefly touch on the risks for men. We know there's an increased chance of prostate cancer. The risk goes up with age. ATM might mean that prostate cancer develops a bit earlier, still rare under 50. But there's 2 options. So one is the profile study, which is being run at Royal Marsden, is a research study though, and you have to go there for that. The other option is to ask your GP for a PSA blood test from the age of 50. Simple test, but it's not perfect. It can be high for many reasons and can sometimes mean you get unnecessary investigations. And the other thing that we know about is a slightly increased risk of pancreatic cancer. We don't have a good screening test for pancreatic cancer, but you may wish to consider joining Europac, who will essentially take your family history, some blood tests and urine samples, and consider whether they think you're high risk. If you haven't got a family history of a close relative being affected with pancreatic cancer, you probably wouldn't be offered the screening through this study because they wouldn't deem you to be high risk. So basically, don't panic. I hope you can see the relative risks of ATM versus lifestyle, for example. When you come up to those big birthdays, think about whether you may be in a position to start some screening or talk about the PSA testing. This is an evolving area, so please don't hesitate to review this with us or with your clinical genetics team on a regular basis. Thank you.
Wonderful. Thank you so much, Anna. Such an important topic, and sorry I kind of barged in
sorry.
towards the end there. But if you have any questions for Anna, do please pop them in the chat. I know there's already one question from Naz. If you're able to have a look at that, Anna, that would be great. Thank you. Okay, so we'll move on to our next speakers. We've got a double act of speakers from the AT Society who are going to be presenting on behalf of Sarah Dernan, who's a dietitian based at the Paediatric Clinic in Nottingham, but Sarah couldn't join tonight, so Anne and Kay are going to present some slides on her behalf, I believe.
Penny's just going to do an introduction, actually, Rob, if that's
Sorry, over to you,
okay.
Penny. Yeah, of course.
Thank you, Penny.
Yeah, okay, so I'm going to try and convey what Kim Chow, who's an excellent scientist who has been working on the role of ATM in metabolism and energy generation. So all the cells in our body require energy, and a very important source of the energy is ATP, which is generated in mitochondria. And you've heard us already mention mitochondria during these talks. And the mitochondria are organelles within a cell. So can you see my pointer? I'm not sure. But anyway, the cell is on the right of the screen with the nucleus in darker blue. Yeah, that's it, darker blue. And then here, and pointing at these little organelles, which are called mitochondria, and they're highlighted on the far right in slightly bigger. And the mitochondria are where the energy is generated. They're the powerhouses, and Kim used this wonderful analogy, they're the powerhouses of cells. And some of his earlier work showed that ATM promotes a pathway that allows mitochondria production to cope with high energy demand. And the next slide, the highlight, I think. Yeah, sorry. There's also evidence that ATM has some other roles in particularly glucose metabolism, which is helping in energy production. So there's impaired energy production in patients that don't have ATM. And the children have a particularly high demand for energy, especially during motor learning. And that's particularly important for the Purkinje cells, which is the cells in the cerebellum which succumb to loss of ATM. So what Kim has found is there is definite roles for ATM in generating energy in cells. Next slide. Yeah. So Kim also talked to us about how 80 individuals have a higher risk of diabetes. And in one study, it was about 50% or more than 50% of 80 patients had high blood glucose levels, and that this diabetes was particularly associated with insulin resistance. And that this was not associated with other risk factors, which often are causal for diabetes, like being overweight, obesity, that sort of thing. This is something that is a consequence of not having ATM. So you've probably all heard about insulin-resistant diabetes, but maybe you don't quite understand what it is. So the next slide, Kim very nicely explained to us how insulin can trigger a pathway to remove excess glucose. And it does that via those little receptors in cells, which are in blue here on the left. And excellent. And that changes this GLUT4 receptor on the cell, which is the one in darker blue, and results in glucose being taken in. He likened it to opening a door and decreasing the level of glucose in the blood, in the plasma. And when you have insulin resistance, as shown on the right, that door doesn't open. So we maintain high levels of glucose in the blood, and that can be harmful. And so there may also be increased glucose in the blood in AT patients because glucose metabolism aberrant, and those then having the increased insulin receptor loss, insulin resistance, makes that blood glucose be maintained even higher. Next slide. So all is not lost, or at least there is hope on the horizon, put it that way. And here there is some studies where ketones and ketone bodies have been used and have been found to be used as an alternative supply of energy. And here Kim likened it to a hybrid car. You can have one way of generating the energy, and you can have another way of generating the energy. And particularly in some, during aging, you can have decreased glucose metabolism. And in that situation, there has been suggestions that ketones and ketone bodies can be used as an alternative energy source. So that's a possibility, for 80 individuals as a way of generating their energy. However, this is still not well studied, and there can be situations where high ketone levels are not beneficial. So at the moment, that is not being recommended to have a sort of ketone diet, but it's work that is ongoing, and that may be a potential hope for trying to improve the energy levels for 80 individuals. So the next slide, I'm just going to give you the major take-home message. The first is that ATM deficiency does affect energy production in cells. There were two other people at the meeting who talked about this, and I think there's really quite strong evidence that that's the case now. And that is especially important in the cerebellum, the Purkinje cells during development. And that, in fact, there was even one talk that suggested that the lack of ability to generate energy efficiently in the Purkinje cells contributed to the progressive ataxia. So the important take-home message here is AT children are not lazy, they're just poor at producing energy. Then AT individuals are also prone to insulin-resistant diabetes, and that's not necessarily because they're overweight but nevertheless, a good diet could be especially important for AT individuals. And finally, there are potential alternatives. We may be able to try and do something about this. And there's evidence that in AT, there's the buildup of toxic intermediates. We may be able to do something about that, but more research is needed. So I'm going to hand over now to Anne, who's going to tell you how you can try to maintain that good diet and deal with this limitation of energy production.
Okay, thanks, Penny. I think we thought it was very important that Penny gave everyone the overview from a scientific perspective. Kay and I are, as Rob kindly said, we're presenting this PowerPoint on behalf of Sarah Durnan because she couldn't be here. Sarah is the clinical academic dietitian at Nottingham University Hospital. She's the dietitian that works within the paediatric AT clinic as well. And just to say, Kay and I are not dietitians, so this will be a whistle-stop tour from a very layman's perspective. Next slide, thanks. I suppose really what we wanted to start off with was to say what Sarah says to everyone is trust me, I'm a dietitian. Everything that we're talking about is a generalised overview of pretty much what we're all asked to follow, you know, from lifestyle that Anna Wilson talked about just now. But particularly when a child is seen by a diet or a young adult is seen by a dietitian, it's actually building up that trusting relationship and actually understanding what their assessment means for that particular child or young person and why they're offering that particular guidance or advice. Next slide.
So, the Eat Well guide, I think we all probably know this. I'm not going to spend long on it, but this is for all of us. This is not just for people with AT. This is not just for carriers of the ATM. This is for all of us, including me, who had a nice croissant at the weekend for breakfast. But just generally looking and thinking what you can replace them with and how that might help going forward. I think, as Yossi said, about taking care of yourselves, as Anna mentioned with cancer, you know, it's for all of us really, isn't it? So we'll be able to show you this in more detail. We'll send you a copy of this so you can see it. Okay, next slide.
This is quite an interesting slide because it gives an overview of research from the current UK eating patterns. So you can see on the left from the Eat Well guide what the recommendation is, percentage of foods coming from fruit, veg, carbohydrates, etc. and the approximate average intake in the UK. And you'll see as you go down the list, when it comes to proteins, it's approximately higher than the recommended amount. Similarly for dairy and alternatives. And then we get to the fats, sugars and salts, and that's significantly approximately higher, as is the oils and spreads. And we'll talk a little bit more about the possible reasons for that as we go through these slides. Next slide.
So adapting this for AT, really, if you have AT or your child or adult has AT, what do we think about? We know that fatigue is such a big issue. We know it is for children and for our adults. So eating little and often is something that we see does help support these children, particularly when they're at school during the day, to manage that fatigue. Eating little and often can really help. High calorie snacks, we'll talk about some examples in a moment of what that might be, what it might look like. Texture can be a problem for some of these young people as well. And then the other side of the coin, I suppose, apart from fatigue management, is weight gain. And we know that weight gain, nutrition in people with AT is difficult. So fortifying the food, additional, again, the high calorie snacks, supplements, etc. Gastrostomy tube feeding is not for everybody, but it is recommended for some of our children. Yeah, but all of this is about the individual child, really, and going back to what Anne said at the beginning, listen to your dietitian and listen to the assessments that are done for your child. Thanks, Emma.
We're not going to go through this in all great
No.
detail. There's a slide towards the end that actually gives quite a nice summary, but it's actually really focusing, as the Eat Well guide states, it's actually looking at trying to, for all of us, to have a really nutrient dense diet, having food that's been the least messed around with by human beings as possible, get it in its natural state, and try and reduce a lot of the unhealthy carbohydrates, in particular this slide is saying. So the things that people are eating: white bread, white rice, sweets, cakes, biscuits, fizzy drinks, all the things that everybody loves, actually isn't that great for us. And another top tip as well is actually, if you are having something that is packaged, have a look at the food labels, take a close look at the food labels and just see what those labels are telling you. Next slide.
And just healthy swaps, really. Just swapping your croissant, as I spoke about, for something, perhaps the porridge or the wheat biscuits there. But actually, they'll fill you up more than the croissant will. Actually, you feel better for having that, I think. So they're just some examples. We'll leave you with them. Next slide. Thanks, Emma.
So again, going back to the fats, again, if you look at some of the food labelling, which can be really mind-blowing, can't it? Because nothing's consistent. But trying to sort of educate yourself really and look at what is an unsaturated fat, what's a trans fat, and trying to reduce the unhealthy fats that often are found in takeaways, processed foods. McDonald's seems to be a real popular thing with a lot of young people we support, and all these hydrogenated oils can be hidden away in these things. So actually just try and check the labels as much as possible. And some of the tips, again, going back to the healthy guide, is looking at trying to introduce fish, oily fish, a couple of times a week, and try and cook things that are grilled rather than fried. It's all simple to say, and it's something we're all needing to do better at, isn't it, really? But we know now, because of the science, there is a particular importance for this, for these young people who have AT.
Yeah.
Next slide.
Yeah, absolutely. And it's not that we know that a few years back, actually, the advice given was for children in school, for example, to have snacks such as chocolate and things like that would give them that sugar and energy boost. So that has all changed with this evidence now that that is not the way to go, because we know that then that could go on to cause, certainly not help with diabetes, etc. So again, looking at a hybrid way of eating, replacing things with the different options. Certainly for energy boost, for fuel, things like the full fat cheese, the yogurts and the milk can absolutely be great, particularly, as say, we notice that with our young children in the school, and looking at different ways to enjoy your food, but replacing it with something else. Yeah. Very easy for us to say, I know. Next slide.
So from Sarah Durnan's perspective, remember this was her presentation. Sarah used to work. She specialised in ketogenic diets, and she felt very strongly that there actually isn't enough solid research to support the use of a ketogenic diet in AT. And the reason she brought this up is because I think some parents have gone to clinics, to children's clinic, and have asked about putting their child onto a ketogenic diet, and she was very strongly advising against it, simply because there's a lack of research. And she talked a lot about her knowledge about what some of the side effects of people when they are using this type of diet. So as you can see there, keto, flu, headaches, nausea, dizziness, muscle cramps, irritability. We've already got these youngsters who are struggling. They're already experiencing differences and changes in their body function. The last thing we want to do is to introduce even more sort of nasty symptoms into their life to impact on their quality of life. And longer term, she was talking about how many people can develop digestive problems, high cholesterol, hypoglycemia, liver issues. All these things that we're now knowing from the research evidence can be a problem separately in AT. So we don't want to sort of exacerbate that by using a poor diet, if that's the case. More evidence is needed, but for the time being, the advice from Sarah would be to not follow this diet, to actually just really focus on a nutrient-dense whole food diet with as much variation as possible. Thanks.
And just in summary, because we want you to have time for questions, so limit the sugary foods. We all know this, but as Anne said, those whole foods, those nutrient-dense foods, the foods that haven't been messed about with, basically, that are not pre-packaged, they're going to be good. They're going to give you the energy. They're going to give you the calories, and hopefully you can enjoy them as well. So yeah, that's us. Thank you.
Thank you.
Wonderful. Thank you ever so much for that. So that's the last of the talks that we've got lined up for you this evening. So we've had a really nice sort of range of talks covering different areas of the current research in AT.